Perfect. Okay, I'll just start then. So yes, thank you very much for this great introduction. So as it was mentioned, this work that I'm going to present today was performed at Cambridge University, where I was a PhD student there. Well, now actually I am at the University of Lausanne doing something similar. So, kind of related to this work, but trying to also encompass the cancer part and the metabolism part as well. So without further ado, I'm just going to move aside the videos so they're not in front of my graphs. And yes, so the things I will present to you today are really focused on the killing response that the T cells, specifically the CD8 T cells, can do when they find a target cell, which is usually a cancer cell or a virally infected cell. So as you can see in this cartoon here, the T cell is able to migrate, find the target cell, and then form a tight immunological synapse, which is this contact site here that you can find in the middle, where then the killing occurs. What's really important about these cells also is that after they induce killing in the target cell, they're able to detach themselves from the dying target and then move on to other targets as well in a process known as serial killing. I'm just going to put on my pointer so it's easier to see. When we look at the site of the immunological synapse here, the mechanism that I will talk about today are going to be mainly relying on the polarization of the lytic granules, which are just modified lysosomes, at the site of the immunological synapse. Here, we have the polarization of the granules and the secretion of cytotoxic proteins. So among these proteins, we can see two of the main players represented in this cartoon. One of them is perforin, which is a protein able to oligomerize on the target cell membrane and create a pore. And through these pores, we're going to have other proteins such as granzymes, one of the main ones being granzyme B, that will then be able to trigger caspase activation in the target cell and therefore trigger target cell lysis. So when I joined the lab, now quite a few years ago, the lab was in the middle of performing a screen that was published a couple of years ago in Nature Immunology. And Philippa Barton was at the time a PhD student. The lab was in charge of trying to understand if they could find novel regulators of T cell killing. So here, as you can see, among more than 500 genes that they looked at, and I should also specify that we looked at CD8 T cells, but other groups in the UK looked at different kind of immune cells as well. She was able to find 11 new hits in T cells that were able to regulate killing. Two of them were mitochondrial protein, and the one that I focused on during my PhD was called USP30. As you can see, represented on the graph on the right, USP30 is a transmembrane protein that is found either on the outer mitochondrial membrane or on the outer peroxisomal membrane. And its main job is to cut ubiquitin chains from specific substrates on either of these two membranes. So you can imagine then that if there are proteins of sparking or PEX2, they want to deposit these ubiquitin chains and therefore trigger mitophagy, so degradation of mitochondria, or pexophagy, so degradation of peroxisomes. USP30 will be there then to cut the ubiquitin chains and therefore counteract this process. In a situation, though, when there no USP30 is present, then these ubiquitin chains will be deposited on these targets, and therefore you're going to have unrestricted mitophagy and pexophagy, and therefore fewer mitochondria and fewer peroxisomes in the cells. What you can see here in a standard killing assay is the reason why we got interested in this protein. The specific killing assay was done by looking at target cell lysis over time. And in blue, you can see the wild-type cells, while in red, you can see the USP30 deficient cells. So hopefully what can you appreciate from this graph is that for the first few hours, the wild-type and the knockout cells were able to kill in a very comparable manner. But after 4 hours or so, the wild-type cells were slowing down, although they did manage to kill all the targets by between 16 to 18 hours. While the USP30 deficient cells stopped killing completely. So this was, of course, very interesting to us because this suggests there was some kind of switch from the ability to kill at the beginning to a lack of killing at the end of the assay. So now I'm not going to tell you all that is known about mitochondria and T cells because otherwise this would take the whole hour that we're supposed to use for our talks. But just for you to know that here I've shown you that this protein was involved in mitochondrial clearance. And just if we talk about the mitochondria being there at all, we know that actually they shift in morphology and composition when we look at different T cell subsets. And we know that just by changing the morphology of the mitochondria, we're also able to skew differentiation on the cells. So that we know that this organelle is very important in T cells. For example, we have very elongated mitochondria that are often found in the memory cells. And because of this very interconnected shape, oxidative phosphorylation is supposed to be very efficient in the cells. While by contrast, if we look at the effector T cells that you see here on the right, the mitochondria around, they're less interconnected. They're often also smaller, and therefore oxidative phosphorylation is thought to be compromised in the cells, or at least not as efficient as in the situation when you have the interconnected mitochondrial network. and therefore these cells are thought to rely more on glycolysis. Now, the three questions that I tried to answer during the course of my doctoral work were, first of all, trying to understand whether USP30 would have the same role in cells of the immune system as it had been described in other cell lines and in neurons. The second one, I wanted to understand exactly how the loss of USP30 could lead to a loss of killing. And then in the third one, I want to understand whether there was a specific mitochondrial function that was actually causing the loss of killing. And we're going to get more about that later. So just by focusing on the first one then, I wanted to know whether a loss of USP30 would also result in an increase in mitophagy and therefore an increase in mitochondrial degradation. So in order to address this question, we use mouse T cells as a model, and all the results I'm going to show you today are going to come from experiments done in vitro. So in this case, we would just get the spleen out of the mouse, we would stimulate the spleen cells in vitro, and then would purify the CD8 T cells. Now, according to this model, I was able to look at mitochondria at different stages. So at the early splenocytes just got from the spleen, and then at different stages post-stimulation. All of this, of course, was by gating on CD8 T cells. Here we have a staining from mitochondrial deep red to just look at how mitochondria were present in the cells. So usually this is used for mitochondrial mass. And what you can see is that in the unstimulated samples, so on day 0, there was a very similar amount of mitochondria between the wild cells and the knockout cells. However, if you follow the red curve, you can see that suddenly after stimulation, we could see there was a loss of mitochondria, or at least not an increase, as we saw in the wild type, in the knockout cells. You can also see actually in later stage of the stimulation, so when we're closer to the contraction phase, this difference was not as big. So it really seems to peak when we have the peak also of the effector phase. We can also look at this by measuring mitochondrial potentials by using TMRE as a dye, and we found a very similar pattern here as well. So in the day 0 cells, so just got out from the spleen without no stimulation, we found that there was a very similar signal in the wild type cells, in the knockout cells. But then after stimulation, so these days corresponds to the ones you see up here, we could say the more the cells were reaching the peak of the effector stage, which is usually between day 5 and day 7 post-stimulation in vitro, the more they appeared to lose their mitochondria. To understand more exactly what was happening, because of course facts can only get you so far, we try to understand what this mitochondria looked like. And here you can see a staining for the outer mitochondrial membrane TOM20, for the outer mitochondrial membrane protein TOM20. And what you can appreciate in the wild type is that there is lots of TOM20 in T cells, and therefore the mitochondria also have this rounder morphology that is known to be very prominent in effector T cells. However, if then we look at the TOM20 staining, but in the USP30 deficient cells, we can appreciate the staining was decreased. We also have quantified this, so that's what you can see on the graph on the right. And we can see that about 50% of the cells have completely lost the TOM20 staining, and the rest has a very, very dim staining, which you can hopefully appreciate in some of the cells here. This was, of course, also confirmed by Western blotting analysis. And here, in the interest of time, I'm only presenting you the results of TOM20, but we did also look at our markers of the mitochondrial matrix and also the mitochondrial membrane as well. Now, of course, at some point, we also asked what's going wrong with the cells. So we do see some TOM20 staining, but we're not really able to discern the morphology as well as we do in wild type cells. So to basically understand this better, we also performed the electron microscopy. And here are some data from Jane Stagecombe, who was a postdoc in the lab when I was a PhD there. And when you look at these images, you can very clearly see the wild type T cells, the mitochondria present themselves as these very electron-dense structures with very tightly organized crystal structure. This does not happen in the USP30 deficient cells. The mitochondria, in fact, are less electron-dense, and the inner crystal structure is almost completely lost. And you have the quantification of this phenotype here on the right. Now, lastly, just to conclude this part, we could see the mitochondria were in fact affected. We could see fewer mitochondria, and the ones that were left seem to be very disorganized. So because USP30 is supposed to counteract mitophagy, we were wondering whether this was due to an increase in mitophagy. And there is a way to measure mitophagy by using this MT-chema probe. The peculiarity of MT-chema is that the emission of this construct will always be around the same wavelength, which is about 633 nanometers. However, its excitation will change according to the pH in which this dye is found. So for example, if the dye is found in an environment where the pH is around 7, then we're going to have optimal excitation by using a 488 nanometer laser. However, if we actually have the probe found in an acidic environment, so with the pH, let's say around 5 or so, then the maximum excitation of this probe will be achieved using a red laser, so just a 561 nanometer laser. So for now, then, I'm going to refer to the alpha mitochondria as the green signal and to the damaged mitochondria as the red signal. If then you put this probe in wild-type T cells, you can appreciate here how most of the mitochondria are green, and therefore they're supposed to be healthy because the probe is found in a neutral environment. You also have some instances on the left about red spots, but these are probably just normal mitophagy that occurs in the cell. However, if you compare this to the data that we can find in the USP30 deficient cells, what you can see here is that there is much more red signal, indicating that many more of these mitochondria can be found inside an acidic environment. And we know this occurs because, in fact, when mitochondria are being degraded, they are found out inside lysosomes. And therefore, if this probe localized to mitochondria to start with, but then was actually shipped to lysosomes, then now will find itself in an acidic environment. And therefore, we would have more of a red signal. All of this together then really told us that indeed the loss of mitochondria that we could see in our samples was due to the fact that mitophagy was increased. And just to show you here some data that I presented to you in the very first slide, we could see that there was a defect in mitochondrial potential in the cells. We'd also try to understand this further by doing just a standard series assay with measuring oxygen consumption. And what we're asking here was whether the mitochondria were as dysfunctional as they seemed from our immunofluorescence and our electron microscopy data. And here's exactly what you can see. So in the case of the wild-type cells, we try to test, of course, the activity of different components of the electron transport chains using different inhibitors. And while the wild-type cells were responding nicely as expected, the USP30 knockout cells were starting very low in oxygen consumption rate from the very beginning, and they were not responding as much to the drug inhibition. So again, indicating these mitochondria are not as functional as in the wild-type cells. Now, the CRS assay has a very nice peculiarity, which is that not only you can measure oxidation phosphorylation via OCR, but you can also able to measure glycolysis. And here we could see that the USP30 knockout cells had more glycolysis going on. And this would make sense because, of course, as they cannot rely on oxidation phosphorylation for the metabolic needs, they're going to need to shift to another pathway, in this case, glycolysis, in order to survive. So then at this point, we're pretty sure that the USP30 knockout cells were having the same phenotype as in other cells in which USP30 was studied, which is a loss of mitochondria, a loss of mitochondrial functionality, as well as mass, of course. And this was all due to an increase in mitophagy. But we did want them to understand really how exactly does the loss of USP30 also result in an increase, in a decrease of T cell killing. And I'm not going to go through all the data here just because it would take way too long, but I'm going to just jump straight to the main results. But just to show you, there are different steps that lead to T cell killing. So we have this initial migration step, which was not affected by the loss of mitochondria. We have the formation of an immunological synapse with the target cell, and that was as well not affected. We then have a signaling cascade that is engaged, well, it's originated by the engagement of the T cell receptor. And we test different both proximal and distal phosphorylation events there to try to understand if at some point this cascade was interrupted, and that was not the case either. And we also measured the secretion of the lytic granules of the plasma membrane of the T cells, which would theoretically release granzyme B and perforin and therefore induce killing of the target cell. And none of these steps seem to be affected by the loss of mitochondria, and specifically in this case, by the loss of USP30. So then I decided to ask myself the question of whether perhaps all the events that were required for killing could occur in the T cell. But then I wondered, could it be maybe that once it gets to the point where it needs to release the content of these granules, perhaps the content of the granules cell is affected, and therefore it doesn't matter if all the steps that are required for killing are still occurring, if the content of these granules is not sufficient or efficient enough to trigger lysis in the target cell. So I first measured this mRNA abundance of both granzyme B, which is one of the main components of the lytic granules, and perforin, and the mRNA abundance was not changed. But then when we looked at protein expression, then we found a difference. You can see in the first Western blot that compared to the wild-type cells, USP30 had way less granzyme B. And then in the second Western blot, you can see that also the amount of perforin was decreased. You can also see the appearance of other bands here that perhaps we can go by later if anyone has a question, that basically just represent the different processing stage of perforin as it is synthesized and then trafficking from the ER to the Golgi and then to the granules, because at each one of these steps, it has a different glycosylation patterns. So that is why it runs differently. So here what you can see then is that the mRNA levels were not affected by the loss of USP30, but the protein levels were. So because, of course, to go from mRNA to protein, we just look at translation, we decide to ask whether there could be some defective translation going on in our USP30 knockout cells. And to measure translation, we were able to use HPG with an analog of methionine. So the way that you perform this assay basically is that you take the cells, you put them in methionine-free media, you let them use all the methionine they have, but just engage in normal protein synthesis. And then after an incubation that can range from like half an hour to a couple of hours, depending on how active your cells are, you can add HPG, and this will be incorporated in the place of methionine as the cells undergo normal protein synthesis. Then at the desired time point, which again can be from just as little as 30 minutes to as long as a few hours, you can fix the cells, permeabilize them, and then label them using click chemistry. And then by looking at the fluorophore, by immunofluorescence or flow cytometry or STS-PAGE, whatever your favorite method is, you can see what's the signal from the fluorophore, and therefore you can also infer how much HPG has been incorporated, and therefore how much protein synthesis has been going on in these cells. Here you can see the results by flow cytometry. So what you can see is that after just an hour of incubation HPG, the wild-type cells were very efficient at incorporating it, and in fact, the majority of them had. But then if you compare the results to the USP30 knockout cells, you can easily see that most of the USP30 knockout cells actually overlap with the negative control, which did not have any HPG at all. So what this means then is that most of the knockout cells have not incorporated HPG and therefore have not been efficiently translating in this 1 hour long incubation that we're doing here. That, of course, is a very striking result because, as you can see, the difference is very, very evident. And it led us to question how this different translation would also affect the T-cell proteome overall. So in order to answer that question, we collaborated with a group of Doreen Cantrell in Dundee, and both Doreen and Julia helped us a lot in understanding more and more of how the T-cell proteome is shaped by the loss of USP30. Here, what you can see in this heat map is just basically the intensity of different peptides. And something that we found really striking at the beginning was that most of the peptides did not seem to be affected by the loss of USP30, which was actually striking because, of course, we saw such a big difference at the level of translation. Then we also want to try to understand which ones were the protein that were either downregulated or upregulated in the USP30 knockout cells. And what you can see on the left then are the proteins that seem to be lost or downregulated in the USP30 deficient cells. And many of them were mitochondrial proteins. This, of course, is something that we expect, not just because of the translation problem that we see in the cells, but really because of the fact that more mitophagy is engaged and therefore mitochondria are going to be degraded, and therefore you're going to find fewer mitochondrial proteins in the sample. However, hopefully what you can see here is that on the left, there are also some blue dots. And these blue dots are non-mitochondrial proteins that are still being found in a lower abundance in the USP30 knockout cells. And surprisingly, many of them were key mediators of the killing response. So we have granzyme A, granzyme B, the cytokines TNF-alpha, interferon gamma, and also the master transcription factor EMs. So then, moving on to try to understand exactly how could all this fit into the fact that the T cells were not able to kill, something that it's important to remember at this point is that the T cell needs to secrete this protein in order to kill the target cell. So then we could easily imagine that after a few killing events, there also needs to be a very strong push towards protein synthesis in order for these granules to be replenished over time. And what's nice here is that we can actually follow these events happening just by looking at the protein by Western blot. So what you can see in this Western blot then, in the first lane, is the amount of granzyme B present in unstimulated wild-type cells, which, as you can see, is quite a lot. Then, if you stimulate T-cell receptor by using an antigen-free antibody, we can see that now the intensity of this band is slightly decreased, so indicating that some of the granzyme B has been secreted. But then also we have the appearance of a higher molecular weight band that we do not find when we treat the cells with cyclohexamide, therefore blocking protein synthesis. So then this must mean that the appearance of this high molecular weight band is a product of de novo protein synthesis. And actually, we know that granzyme B, when it's first synthesized, is a few more amino acids longer than its mature form. And therefore, the upper bar that you can see here, then it's de novo synthesized granzyme B, which then after a cleavage event is stored into granules in its mature form. If now you look at the same pattern in the USP30 deficient T cells, what you will find is that there is less granzyme B to start with, as I've shown you before. And then after stimulation of the T-cell receptor, most of this granzyme B has been secreted, but there does not seem to be an equal push for the synthesis of granzyme B in order to refill these granules over time. So really here, what we think is happening is that these cells are able to kill at first because they do have some of the killing proteins to start with. But because of the defect in translation, they're not able to resynthesize them and therefore to sustain killing over time. Now, to address this question directly, we then ask ourselves, if protein synthesis is so important for T cells to kill, can we just take T cells, treat them with cyclohexamide, and see what happens? And that's exactly what we did here. So again, we took the wild type cells either untreated, which you can see here in treated with DMSO, or treated with cyclohexamide. And for the first few hours, these cells kill in a very comparable manner. However, after a few hours, the wild type slows down, but still manages to kill over a few hours. But then the cyclohexamide treated cells stop killing completely. And if you remember the graph I showed you at the beginning, which I'm just going to present to you again here now, this pattern of the loss of killing occurs around about the same time as the time that the USP30 T cells stop to kill as well. So these results really strongly indicate to us that the lack of protein synthesis is really, or at least was very likely to be, what was causing the killing defect in the USP30 knockout cells. And just to conclude my talk, I will just tell you a bit more about the different mitochondrial function that we investigated here. Because of course, something really important to remember is that the USP30 model is a model in which mitophagy is triggered into a not very physiological way, let's put it this way. So you have like a clear degradation of most of your mitochondria, and the ones that are left are not functional. But we wanted to understand whether defect in killing and the defect in translation was due to the fact that a whole organelle was missing, or whether it could be pinpointed to a specific mitochondrial function that was required in order for killing to occur. And just to show you a few things that we tried, although I'm not going to go into them now, we know, of course, that mitochondria are important for many different processes in the cells. So we were able to test calcium flux, mitochondrial translation, the TCA cycle, and also we were very lucky to be working closely with the mitochondrial biology unit, which is also found in Cambridge, so that we're not only able to use different pharmacological treatments to address each one of these pathways, but we're also able to use different mouse models. And for today, I'm just going to focus on the results that we obtained with mitochondrial translation. And something really important to see here is that I showed in the first few slides that we can block protein synthesis in the cytosol by using cyclohexamide. But actually, we can do the same in mitochondria by using other drugs, such as doxycycline or even chloramphenicol. So the data I will present to you here, just to finish off my talk, are going to be the ones obtained with chloramphenicol, but we obtained the exact same data also using doxycycline. And what we could see was that just by treating wild-type T cells with chloramphenicol, we could see a dose-dependent decrease in killing over time, showing us that the more mitochondrial translation was inhibited, the more we could observe a loss of killing. Then we decided to ask whether cytosolic protein synthesis was also affected by blocking mitochondrial ribosomes. And we measured again HPG incorporation, and in this case, of course, the fluorescence that we can see by flow cytometry, to ask whether cytosolic protein translation was also affected. And as you can see here, that was indeed the case. So then to ask really the killer question, we ask, okay, so we have a defect in killing, we have a defect in protein synthesis. What's happening to proteins such as granzyme B? Are they being resynthesized to refuel the granules? So if you look at the first part of the graph, as before you have the amount of granzyme B in untreated cells, then the trigger of the T cell receptor, which also triggers synthesis of de novo granzyme B, and then of course secretion of the pre-synthesized granzyme B as well. But then if we look at the appearance of this de novo synthesized granzyme B in the chloramphenicol treated cells, we can see that now this synthesis does not occur anymore. So overall then, we can see that just by blocking mitochondrial protein synthesis, we were able to recapitulate the results that we observed with the USP30 knockout cells. So to finish off, then what I'm showing today is that by having a USP30 knockout, we also can see that the mitochondria are lost and T cell killing is inhibited. We were able to try and see that this was due to the fact that there were fewer killing proteins after sustained killing. And the reason why this was happening was because there was a defect in protein translation. At the same time, we're also able to pinpoint the fact that inhibiting mitochondrial translation is sufficient to also block cytosolic translation and therefore affect the levels of the cytolytic proteins that are required to kill. So with that said, I would just like to take a few moments to thank all the people that were with me in the Griffiths lab, which again is where this work was performed when I was at the University of Cambridge. Of course, our collaborator in Dundee, such as Doreen Cantrell, and then all of our collaborators at the Mitochondrial Biology Unit. And of course, none of this work would be happening without the people working in the facilities here. So I would also like to take the time to thank Dundee Proteomics Facility and the flow cytometry and microscopy facility at the Cambridge Institute for Medical Research. And that said, we'll be happy to take any question.
Thank you very much, Miriam. And yes, I hesitate the audience. If you have any question, please type in the chat or raise your hand. I could give you the microphone. We have one question in the chat from Ophelia, and she asked, how is USP30 function in the tumor microenvironment in these CD8 T cells?
Excuse me, could you repeat? How's the USP30 function?
In the tumor microenvironment.
That's a very good question. It's a very good question. So all of the experiments here were done in vitro. So we don't know whether actually that will recapitulate in a mouse model of tumor or, even like in human cells. We don't know that so far. It's a bit difficult, sometimes to know these things because, USP30 being a mitochondrial protein, most people that have this kind of mutations get referred to, the mitochondrial clinics because they tend to have defects, for example, at the levels of neurons, heart tissue, muscle. So sometimes, it's difficult to try to make a statement about what would happen with these mutations, for example, in humans, because these people will not go to immunology clinics. So that's the reason why I cannot tell you whether, for example, it is known whether patients with this mutation have more or less of a response in the case of a tumor. And because we did not try this in mice themselves, we do not know what will happen, but definitely would be really interesting to try this.
We have another question from Chun Wei Shen. A very interesting talk. Does CTLs have different expression level of UCP30 under inflammation? One paper mentioned that PINK1 and parking inhibit mitochondrial antigen presentation. So I'm wondering if UCP30 will impact cytotoxic T cells as UCP30 has been shown to interact with parking.
It's a really good question. I think we're entering in a field where I'm not an expert, actually. So I think, correct me if I'm wrong, but if I understand the question correctly, when you're talking about the antigen presentation, and now you're not talking anymore about the T cell, you're talking really about the cancer cell as well. So I think, in this case, if I were to imagine, let's say, a compound situation where the USP30 is knocking T cells and there's some problem with PINK1 in the tumor cell, it's a bit hard for me to tell you what the end result would be. It's definitely something interesting. It is known that autophagy is regulated very tightly in the cancer cells, for example, to provide more nutrients or, to downregulate antigen presentation. So, but I think here now we're really going to feel that it's not my field of expertise, which is, the in vivo work.
Okay. And I will have just two last questions. The first one will be, have you tried to, it was a total knockout animal, right? The UCP30?
Mm-hmm.
Yeah, but what about UCP30 function on memory T cells or on these T cells that are, stem-like cells? If
Mm-hmm.
the dynamics of this protein is similar or is just different?
I see. So as I mentioned, this big screen that my previous lab participated in was on a different subset of the immune system. So as far as I remember, I don't think there were any functional issues or developmental issues that were found in B cells. But if you go back to the publication, I'm sure you can find all the data because we were not involved in B cell work directly. In terms of memory cell formation, so when I took the spleens, where I was taking my T cells from, all my staining started with looking at the naive versus effector versus memory population to see whether differentiation will be affected. Because we do know that actually by having different mitochondrial dysfunctions, we can also affect the differentiation of T cells into different subsets. In our case, the memory T cell formation was not affected. But of course, it would be nice, for example, to take these cells, stimulate them, wait for them to lose the mitochondria, and then maybe also measure in vitro whether we can see the appearance of markers associated with like a memory pool formation. Another thing that I would like to mention is that if I just go back to one of my first slides, let me just go here. Something that I think we found very interesting. So when I was doing the staining for different mitotrackers here, what you can see is that really the biggest defect happens at the peak of the effector stage, so between day 5 and day 7. And actually, by the time you get to day 9, so to later days after stimulation, the signal that we found both for mitochondrial mass and mitochondrial potential was very similar. So in this case, what we mean, probably, we don't know it yet. We don't know whether the cells are somehow compensating the lack of USP30 and therefore they're regaining the mitochondria, or we don't know perhaps whether, as you can see here, for example, in this curve, whether these cells with a very low TMR staining maybe are dying, and therefore what we end up with at the end are only these cells here of the higher part of the curve. They're the ones that did manage to conserve some mitochondria. So it is possible that the cells that do survive are the ones that were doing better to start with, and therefore that's why by the time you get to the memory T cell pool, you do not see like many differences there. That could be one of the reasons.
Finally, my final question, there was a paper from Erica Pierce's lab in the same topic saying that fever induced protein translation in T cells and induce cytotoxic killing. Do you know if this mechanism is related with the one maybe UCP, fever induces something with UCP theory or something related to mix the stories, or it's just different stories there?
So the stories are slightly different, but I think they're converging really towards the same idea. The idea here, if I remember the paper correctly, was that by having a fever model, they also saw more mitochondrial mass. And therefore, they were trying to link the fitness then of these mitochondria to the fact that T cells were better killers. So I think, overall, the two papers support each other because they both identify, again, if I'm not wrong, mitochondrial translation as, a key function for T cells to be able to kill. I think the main difference is that in their case, they tried to check whether there was a connection between mitochondrial translation, cytosolic translation, and they did not believe that that was the case. So that, of course, was very interesting to us because actually when that paper was published, I was just in the middle of publishing mine. So, you also ask yourself, okay, so what's the difference here? Of course, the model was very different from ours. That could be one of the reasons. And the other thing is that actually there are many ways that cellular stress can be relied and block protein synthesis. So there are many ways that mitochondria in translation can communicate with the cytosolic machinery, but there's also like many ways that just stress in general can block protein synthesis. So I think, the two papers support each other in saying that mitochondrial translation is important. Now, exactly how it feeds then to the killer response, I guess, depending on the stimulus or depending on the insult that you have on the cell, that could be happening via a variety of different pathways.
Well, thank you very much, Miriam. And yes, and we can move on with the next talk. Congratulations.
Thank you.
Yeah. So, Dr. Peng, if you could share your slides, please. Thank you. So I will introduce Dr. Peng. Dr. Peng did his PhD in biomedicine